All right, we're back and ready to dive in. Today we're tackling a topic that you, our listener, requested: advanced mass spectrometry. We've got some seriously technical material to work through today, so buckle up. We're going to be looking at FTICR and Orbitrap technology, and then we'll see how tandem mass spectrometry takes analysis even further. Basically, it's all about getting a much more precise and detailed look at the molecules in any given sample. And luckily, we have an expert here to help us break it all down. Welcome.
Glad to be here. Yeah, these techniques are really amazing. I mean, they really highlight just how far mass spectrometry has come. They let us analyze molecules with amazing precision.
I'm excited to have my mind blown. Now, I know that the basic principle of mass spec is separating ions based on their mass-to-charge ratio, but things get a lot more complicated with FTICR and Orbitrap, right? What makes this technique stand out?
Yeah, for sure. They take mass spectrometry to a whole new level, both in terms of resolution and accuracy. Let's start with FTICR, which stands for Fourier Transform Ion Cyclotron Resonance. It's all about looking at how ions behave in a magnetic field.
Right. I'm looking at this Mass Analyzers to Essity 2024 document right now. It says that ions get trapped in a magnetic field, and their movement, or cyclotron frequency, is related to their mass-to-charge ratio.
Exactly. FTICR uses something called a Fourier transform to analyze the signal from all those ions swirling around. And this allows for really precise mass measurements. Think of it like going from a blurry photo to like a high-definition image.
Oh, okay, I see. So stronger magnetic field means a clearer picture of those molecular masses, kind of like a more powerful lens on a microscope.
You got it. And that's one of the big reasons why FTICR is so powerful, especially for analyzing complex mixtures like what you find in proteomics research.
Makes sense. But the document also mentions other things that can impact FTICR accuracy, things like electric field homogeneity and phase correction.
Right. Those are important for making sure that the ions behave predictably and that the signal is interpreted correctly. It's kind of like calibrating your microscope before you look at a sample.
Got it. So it's all about fine-tuning for that ultimate precision. FDICR sounds incredible. But we also have the Orbitrap, another contender in the high-resolution game. I see some interesting visuals here about its history and how it works. So how is it different from FTICR?
Well, FTICR relies on a super strong magnetic field,
Hmm.
but the Orbitrap uses an electrostatic field to trap the ions. So they orbit around a central electrode, and their oscillation frequencies, much like an ICR, reveal their mass-to-charge ratio.
Okay, so different forces at play, but the same goal: pinpointing those masses. How do they stack up against each other in terms of applications and limitations? Is one just better than the other?
It really depends on what you're trying to do. FTICR generally has better resolution and mass accuracy, but it's definitely more complex to operate and more expensive. The Orbitrap, though, is more compact and affordable, so it's more accessible to labs that maybe don't have the resources for FTICR.
So it's like choosing between a super high-power sports car and a reliable sedan. Both can get you to your destination, but the experience and cost are quite different.
That's a good analogy. The choice really depends on the complexity of your sample. For really complex mixtures where you need to ID and quantify thousands of molecules, the resolving power of FTICR might be essential. But for more simple applications, the Orbitrap could be just fine.
So choosing the right tool for the job makes sense. I'm also curious about tandem mass spectrometry. From what I understand, it's not just about measuring the mass of a molecule. Can you explain how that works?
Sure, Tandem MS or MSMS takes mass spectrometry a step further. We don't just measure the mass of an ion, we actually fragment it and then analyze those pieces. This gives us super valuable insight into a molecule structure, and it helps us identify unknowns.
Oh, wow. It's like taking apart a puzzle to see how all the pieces fit together. I read about two main approaches: tandem in space and tandem in time. What's the difference?
Well, Tandem in space uses multiple mass analyzers, one after another, and each one has a specific function. The classic example is the triple quadrupole mass spectrometer. Ions are selected, fragmented, and then analyzed in separate stages.
So it's like an assembly line for molecules, each station doing its own thing. I see a diagram labeled tandem in space that shows this. What about tandem in time? How does that one work?
So in Tandem in time, it all happens in one mass analyzer. Ion traps and ICR instruments are really good at this. They can trap ions, fragment them, and analyze the fragments all in the same space.
Wow. It's amazing how they can juggle all of that. So how are these ions fragmented? What is that process like?
One common method is collision-induced dissociation, or CID. Basically, we make the ions collide with neutral gas molecules. They absorb energy and break apart into smaller pieces.
Oh, okay, so it's like a microscopic demolition derby. But the debris actually tells us a lot about the original molecule, right? Because each one breaks apart in its own unique way.
Again, it gives us a kind of fingerprint that we can use for identification. And we analyze these fragments using various tandem MS scan modes. Each mode is designed to answer a specific question about the molecule.
Right. I see a diagram here called pictogram symbols for tandem MS experiments, which seems to illustrate these different modes. What are some examples of these modes?
Mm. Well, in a product ion scan, we pick a specific precursor ion, and then we analyze all the fragments it makes. This tells us about the structure of that ion. And then we have the precursor ion scan, which is basically the opposite. We select a specific fragment ion and look for all the precursor ions that made it, which can help us find unknowns that share a common structural feature. And then there's the neutral loss scan, where we look for precursor ions that lose a specific neutral fragment, which can be helpful when identifying molecules that belong to a particular class.
Wow, it's like having a whole arsenal of tools for molecular detective work.
Hmm.
We can pick and choose which scan mode we want to use to pinpoint a specific molecule.
Or a structural feature, depending on what we're looking for.
Exactly. By combining these approaches, we can get a really detailed picture of the molecules in our sample, their structures, and even how they interact. That's incredible. So much information just from tiny little pieces. Well, before we dive even deeper and talk about specific examples of these fragmentation patterns, let's take a quick break. We'll be back in a moment to see how understanding those patterns helps us understand molecular structure. Stay with us. Welcome back. So before the break, we were talking about how tandem mass spectrometry lets us fragment molecules and then analyze those pieces, like a molecular jigsaw puzzle. I'm curious how we actually make sense of all those fragments. It seems like you would need a lot of expertise to interpret those fragmentation patterns.
Oh yeah, for sure. It does take a lot of knowledge. But just like a detective, they look for patterns and clues at a crime scene. Scientists use what we already know about how different molecules break apart to help make sense of the data from the mass spec.
Oh, okay. So it's like having a cheat sheet for molecular mysteries. We know how certain types of molecules tend to break apart, so we can use those patterns to ID the culprit.
Exactly. So for example, let's look at a molecule with a benzene ring. Benzene rings are pretty stable, but they still fragment in predictable ways.
Right. It looks like this source material shows that the fragmentation often happens at the benzylic carbon, which leads to a resonance-stabilized benzylic carbocation, which then rearranges to form a tropylium ion.
Yeah, exactly. And that specific fragmentation pattern with the tropylium ion is a strong indicator that you've got a benzene ring somewhere in your molecule.
So we can use these patterns to differentiate molecules even if they have the same mass.
Yes, precisely. That's one of the huge advantages of tandem mass spec. We can actually tell the difference between isomers, molecules that have the same formula but different structures, by looking at how they fragment.
That's super useful. It looks like the material that our listener sent covers a lot of different fragmentation pathways for like alkanes and alcohols and ketones and more. Should we go through each of those? Or are there just a few that would be more helpful for getting the big picture?
Maybe let's focus on a few key examples that highlight the main concepts. Like, alcohols tend to lose water when they fragment. That's really characteristic of that class of molecules.
Right. This example with 1-propanol, the mass spectrum clearly shows a peak that corresponds to the loss of water.
Exactly. And knowing this can help us quickly figure out if we might be dealing with an alcohol. Ketones are another interesting case. They often break apart to form what are called acylium ions,
Okay.
which are really characteristic of ketones.
So we'd be looking for those acylium ion peaks as a clue that we might have a ketone.
Exactly. And the key is knowing what these fragments tell us about the original molecule. It's not enough to just identify the pieces.
This is so cool. It's like a secret code that unlocks the language of molecules. So let's talk applications. I can imagine that knowing how to read these molecular fingerprints would be useful in so many fields.
Oh yeah, definitely. One of the big ones is identifying unknown compounds. Like, let's say a chemist makes a brand new molecule, or someone finds something mysterious in a sample. Tandem MS can help figure out its structure.
So like a molecular detective, you feed the mystery molecule into the mass spec, look at the fragments, and compare those to known patterns.
Yep. And you only need a tiny bit of sample. We can ID unknowns even from just a few nanograms.
That's incredible. What other applications can you think of?
Proteomics is another big one. The study of proteins.
Right. Proteins are so important for everything. Understanding their structure and function is key for so much research.
Yeah. And Tandem MS allows us to actually break down proteins into smaller peptides and then figure out their amino acid sequence. This helps us understand how they work, ID disease biomarkers, even develop new drugs.
Wow, that's like taking apart a complex machine, figuring out how all the parts fit together.
Yeah, and the level of detail you can get with these instruments is
Yeah.
amazing. We can ID and quantify thousands of proteins in a single experiment.
That's wild. So we've got IDing unknowns,
Yeah.
analyzing proteins. What else?
Metabolomics, studying all the small molecules in a biological system.
Right, the building blocks of life.
Exactly. By looking at the metabolites in a sample, we can learn about the metabolic processes happening in a cell or an organism. This can help us diagnose disease, see how well treatments are working, even personalize medicine based on someone's metabolic profile.
It's like taking a molecular inventory of the body.
Yep.
What else?
Oh, environmental analysis is a big one. We can use Tandem MS to detect and quantify pollutants
Okay.
in water, soil, air. That helps us monitor and protect the environment.
A molecular watchdog for the planet.
You could say that. And this information is critical for cleaning up contamination and preventing pollution.
So important. And I imagine food safety is another one, with all the concerns about contaminants in our food.
Yeah, definitely. Tandem MS can be used to detect all sorts of things in food. Pesticides, herbicides, antibiotics.
So no more fake olive oil or mystery meat. We can eat with confidence.
That's the goal. As concerns about food safety grow, Tandem Mass Spec is only going to become more important.
It's amazing how this tech has an impact on so many parts of our lives. We've talked about IDing unknowns, analyzing proteins, metabolomics, monitoring the environment, food safety. Is there anything it can't do? What are some of its limitations?
Well, it's not a perfect tool. Sometimes.
Yeah.
It can be tough to figure out the structure of a molecule just based on its fragmentation pattern, especially if you have isomers that produce very similar fragments.
Right, so going back to the detective analogy, we could have two suspects with matching fingerprints. What do we do then?
In those cases, we often have to use Tandem MS along with other techniques like NMR or X-ray crystallography to get the full picture.
So not a standalone solution in every case. What other limitations are there?
Well, sensitivity is a big one.
Oh.
Tandem MS is really sensitive, but it still needs a certain amount of sample. If you're looking at trace amounts of something, that can be hard.
Like trying to hear a whisper in a really loud room.
Exactly.
You mentioned matrix effects earlier. Can those be a problem too?
Oh yeah, sometimes.
Yeah.
The other stuff in a sample can interfere with ionization or detection, and that can mess up your results.
Got it. Like trying to find a needle in a haystack. But the haystack keeps getting in the way.
Yeah.
Anything else?
Data analysis is a big one. Tandem MS makes a ton of data, and making sense of it all can be tough. You need special software and expertise.
It's one thing to have all the puzzle pieces.
Yeah.
But you need to know how to put them all together.
Exactly. As this technology keeps getting better, bioinformatics is going to become even more important.
That's fascinating. So Tandem MS is a super powerful tool, but it's good to know its limitations and use it in the right way, often alongside other techniques. Well, we've covered a lot, from how molecules break apart to all the different ways Tandem MS is used and what it's good at and not so good at. Before we wrap up, I want to hear your thoughts on the future of this technology. What's next? What are some of the things that researchers are most excited about?
Well, it seems like mass spectrometry has come a long way, and it's still evolving. What are some of the things driving that innovation? What are the big questions or challenges?
One area that's really exciting is sensitivity. Developing instruments that are even more sensitive. Being able to detect and analyze molecules at even lower concentration.
Wow, that would be amazing. What kind of impact could that have?
I mean, think about it. It could completely change things in fields like medicine and environmental science. Imagine being able to detect single molecules or analyze a single cell. That level of sensitivity would open up so many possibilities.
So instead of looking at like the average composition of a tissue sample, we could zoom in and see what's going on in individual cells. Yeah. And in environmental science, we could detect tiny amounts of pollutants much more accurately, and that would help us identify problems and address them more effectively. Plus, we could track pollutants through ecosystems much better.
Wow, so many possibilities. What other advancements are you excited about?
Well, there's also a lot of work being done on new ionization techniques. Remember, ionization is that first step in mass spec, where we give molecules a charge so we can work with them.
Right, it's like giving those molecules a backstage pass to the show.
Yeah.
What are some of the challenges with the current ionization methods?
Well, some molecules are really fragile, and they can actually break apart during ionization. which makes them really hard to analyze.
Yeah.
So scientists are trying to develop gentler ionization methods that can handle those delicate molecules.
So it's like figuring out how to invite those shy molecules to the party without scaring them off. What kind of molecules are we talking about here?
We're talking about big biomolecules like proteins and DNA, and things like complex carbohydrates and lipids. They're really important for biological processes, but they've been tough to study with mass spec because they're so fragile.
So if we can figure out how to ionize them gently, that unlocks a whole new world of possibilities.
Exactly, and that would have huge implications for drug discovery, disease research, even material science.
Incredible. It seems like mass spectrometry is always pushing the boundaries.
It really is. As we get more sensitive instruments and more gentle ionization techniques, who knows what we'll discover?
This has been a fantastic deep dive into mass spectrometry. We've covered some pretty complex stuff, but hopefully in a way that's fun and engaging.
I think so. It's been great talking about all this with you.
And to our listener, thank you so much for suggesting this topic. It's been challenging, but really rewarding. We've learned about FTICR and Orbitrap technology and how they give us such high-resolution analysis. We've explored tandem mass spectrometry and how we can use fragmentation patterns to understand molecules, and we've even gotten a glimpse into the future of this technology.
Yeah, it's a good reminder that there's always more to learn.
Exactly. So keep that curiosity alive. Keep asking questions. Who knows, maybe you'll be the one to make the next big discovery. Until next time, keep exploring.